Okay, so this is a presentation that I would normally want to do in person and live as we're talking about what to do for the bioinformatics and informatics project, but we'll manage here. So I wanted to give you a little bit of background. What you're going to end up doing, of course, is let's see, come on, where's the little problem here with my streaming software. Sorry about that. You just saw my little head, I think. There it is. Okay, come on. Where are we? There we are. Okay, so let's try to move forward on the slide. Okay, so anyway, what you're going to be doing, and hopefully you've been working on this, is that you have either started or completed some work with your gene trees. So I had asked you to, complete your sequence alignments and to generate your neighbor joining trees, maximum likelihood trees, etc. And so you can go ahead and just work with the neighbor joining tree. But two things that we want to do is we want to see how well our gene tree fits what we would expect. And so what we would expect is basically what's called a consensus tree. So that is based on lots and lots of different traits, not just your single gene. And so we have lots and lots of evidence to support the bifurcating tree among our 14 species, including humans. And so to see how well our gene trees estimate the true phylogeny, we can do two things. And so these were processes that I asked you to go through. One of them is called a gene tree reconciliation, which is basically comparing your gene tree versus the consensus tree. So that was a Lab 6 working with gene tree reconciliation sheet. And your results are supposed to go into your digital notebook, and you would use that as part of your bioinformatics project. And so as we go through these slides, I'll point out where that fits in. Then the second thing that I'm asking you to do is to calculate rates of evolution along the tree. So taking two species at a time, comparing it to an outgroup, they should have the same distances, and if they don't, that means that there's different rates of evolution going on, and that's something we need to know, because the whole point about this is to talk about a particular model of evolution called the molecular clock. And so this is the talk that we're giving here, where it says slides molecular clock. And what you would want to do between today and into next week is to actually use your NUIC files to pull out information about the genetic differences between each pair of species. Those are your estimates of the amount of genetic differences between them. And so off we go, right? And so the build your molecular clock is the main thing that you're going to do, and then there's a couple of help files or help pages in your canvas about how to get the distances from a distance tree, and then how to work with the pairwise divergence times. Okay, so at the end of next week you're going to want to be submitting your molecular clock files. All right, so on to some background. So, look, we've talked about this, your genome, and if you're a mouse, if you're an alligator, or if you're a human, your genome is a reflection of history. And so we can trace our history, our genetic ancestry, through, of course, changes in DNA sequence. And it's a pretty messy and mathematically and complicated endeavor, but if it's employed in the correct way, people are horny. That's not what it's supposed to say. Lives are complex. The secret history is truly hidden in the mosaics of our genomes. But caveat emptor. Actually, this is a blurb from this book, so it's kind of funny. All right. I hadn't even noticed that. Oops. Okay, so remember what's in your genome. Our genome's a little over 3 billion base pairs long. Lots and lots of different elements, okay? Transposable elements, viruses, pseudogenes, functional genes, non-coding genes, protein coding genes, etc., etc., centromeres, you name it, right? So quite a bit of it is conserved, some of it is not. So that means comparing it to chimpanzees or comparing our genome to other primates, a good 26% of the DNA between genes is different, right? Not conserved. So that's something we can use as we're reconstructing phylogenies, the parts that are shared and the parts that are not. Okay, so genomes, basically if you think about it, every species has its own genome. There's variation among individuals, populations, individuals, but genomes for chimpanzees, that's what you get, a chimpanzee. All right. So Darwin and others before him recognized that all species have an inherent capacity for exponential growth. So this is, following bacteria growth on a petri dish over time, and what you're supposed to get is this idea that every species has the capacity to increase like this. All right? Okay. But species don't continue to increase exponentially over time. There's limits, and so within those limits the population size remains more or less stable, but that doesn't mean that everybody has an equal chance of surviving and leaving kids. And so there's two forces, if you will, that explain diversity, genetic diversity at any particular time. There's both chance, so genetic drift, some individuals leave kids purely by chance, and others leave kids because the environment changes in a particular way and they are the only individuals who have the right stuff. So these two processes, natural selection and genetic drift, sift through populations, and some have then resulting in some leaving more kids than others. Okay? Speciation then is the accumulation of those genetic differences, but requiring some sort of a barrier keeping individuals from having kids together. So reproductive isolation is a key component to this. But as long as you can separate two populations, and within each population, individuals are able to survive and have kids. But over time, those lineages are independent of each other, and so they accumulate differences through mutation. Right? And over time, the idea is that we bring them back together, remove the barrier, and they can no longer breed together. And so that's the full speciation event. So barriers can be physical, like the Kapuka lava flows. And so Hawaii is famous as an incubator of evolution. For example, those little fruit flies that we swat away, there's hundreds and hundreds of species of these guys on the islands of Hawaii that they're found nowhere else in the world. And it's in part because of this process of lava coming in, separating islands of habitat from other such islands. And over time, then the populations can differentiate into new species. Over evolutionary time, of course, continents have not been where they've always been, and so this is an animation of the history of the continents. So way back when there was Pangea, just one, and of course now there's multiple continents, as we know. So anyway, there's evidence left in the fossil record of these events. So for example, there are camels in South America, and there's camels, of course, in Africa, and they're more closely related they are than to other such critters within the continents, indicating a land bridge or, again, the adjacent continents. And so this is a mapping of various species that you could go. We find them in South America, Africa, India, whatnot, the fossils. And so the best hypothesis is that at some point, of course, the continents shared land bridges with each other. And of course, this has now since been corroborated by multiple types of information. Okay, so similarity is amazing. Mice and humans share about 70% sequence identity. Humans and chimps, 98%. Okay. Branching order identifies monophyletic groups. And so I've got up here the lizard, alligator, and chicken, right? And so those form the reptile groups. Here's the opossum, that's our marsupials. And then here are our placental mammals, including chimpanzees, dogs, cats, and humans, right? Okay. And so the idea is we can, for any number of species, we can, using fossils and morphology, evidence through development, and of course DNA and protein sequences, we can come up with what's called a consensus tree. This is our best guess as to who's related to whom. And the further back away we go from the tips, of course, that's further back in time. And if we can go ahead and root the tree, which we would do where you see the bifurcation from opossums to the reptiles there, all right, this gives us the direction. So in groups are placentals, out groups are reptiles, all right, and it gives us direction of change. That's an important part of constructing phylogenies. Okay, so differences are nested consistent with coalescence. So the idea that if you start from any number of groups at the present and then you follow things back in time, you're all going to converge on, in effect, the same set of populations. And so this is a little simulation where randomly we allow just a handful of individuals to have kids, and if we follow for 10 generations, okay, at the end of it we'll find that everybody traces back to just one group, right? So everybody else failed to have kids over time. Okay, and this is the formula for this coalescence. All right. All right, so the results of that was basically only the J group survived. Okay, so anyway, this is this fundamental coalescence, this idea of sort of nested relationships going back in time, is what 23andMe and Ancestry.com use when you send in your sequences, and they gather up your single nucleotide polymorphisms, et cetera, in particular what are called haplogroups on the mitochondrial DNA or on the Y chromosome. And so we can then build trees based on this and relate the diversity of those haplogroups to geographic ancestry. And so assuming that the databases have equal representation from all parts of the world, and those parts of the world are well sampled with folks that live there as opposed to have migrated there, you can come up with these sort of fuzzy math things that say somebody is like 17% Irish and 92% something else, right? Okay. All right. So the coalescence model, though, assumes that you don't have any gene duplication. If you do, that then means things will look less similar, meaning you'll have greater differences than you would have thought before. And you also then, when you're building your ancestry, you want to have the genes not linked together. So, okay. All right, so just as a review, similarity between two taxa, what we're talking about is homology, right? So homology is not just similar, it means similar because shared ancestry. So this is a diagram of showing the difference between analogous, two unrelated forms converged on the same solution, okay, versus homology in the sense of you're the similar because of shared derived ancestry. Okay. All right. So given some sequences, how can you build a phylogeny from it? So I've got 3 species and just a short amount of sequence, and species A has a single nucleotide differences. So what's the simplest phylogeny can you construct? All right. Well, you would say probably species B and C go together, and species A is perhaps the ancestor. So that's one. But what's the most likely ancestor sequence? All right. Well, because we have two that have the C, we think that the ancestors must have C also, and therefore the derived condition is going from C to G. Okay. Now, if we have additional data, we can then test to see whether or not our simple tree is the correct one or not. So if this was our ancestral sequence, and we now have, this could have been the possibility. Okay. And so we can then reconstruct the different possible ancestries. And so the trick is, though, is that we won't be able to tell this, right? So if in the end we have these two sequences, and if this was actually the ancestral sequence, okay, that mutational step is lost to us. So that's why we say when we estimate phylogenies, of course, that they are sort of minimal estimates of the amount of evolutionary change that occurs. A great deal of the change going from A to D, A to T, G to C, et cetera, while you can have repeated evolution going from A to T and then from T back to A, right? And so you would miss that A to T, that first mutational event, because in the end all you have is A. Okay. So again, gene trees don't always reflect the true phylogenetics, and that's because of this sort of repeated evolution thing. Character reversal, yeah, that's what I wanted to talk about, right? So if character reversals occurring in your gene sequence, you won't be able to tell directly, and the neighbor joining algorithm, the maximum likelihood algorithm, will in effect combine the sequences based on your sequence alignments into the best grouping that they can get statistically, but that doesn't necessarily mean it's the true phylogeny. Okay. So in principle then, you need to keep track of the ways genes can be homologous. And so genes can be homologous and then be orthologs if you inherited the gene directly from another species, right? You know, your ancestral species. And those are the ones that we think that we're working with in the lab through our blast searches and whatnot. But we haven't done additional tests to confirm that, so it's very likely that we have sequences that are similar, but they're similar because they're paralogs. In other words, we've grabbed genes that are the result of gene duplications after a speciation event, in which case we can't tell the difference. We'll be comparing them, but it will lead to incorrect estimations of phylogeny. The gene tree is fine, but it can differ from the true phylogeny. And then xenologs we won't really talk about, but xenologs are the idea that it has literally been inherited from another species directly. So, so like viruses bringing in genes or something like that. Okay, so we've already just talked about orthologs, paralogs, and xenologs. So these are your definitions. Orthologs, two genes A and B, similar because two shared recent ancestor. And again, orthologs are what we hope we have collected in our lab exercises. Okay, so in lab 6 exercises, I asked you to carry out an analysis, what's called tree reconciliation. And it's a pretty simple idea. Basically what you do is you take your consensus tree, which is built on lots and lots of data, and then you compare it against your gene tree. And you basically see, well, how many of the nodes, so those branches, right, how many of those nodes match up? So this is from program R software, and so in lab 6 I provide a whole bunch of scripts that you could use to run and recreate this for your own gene. Okay, and so in the end what we have is we have 1, 2, 3 different nodes, 4 different species traceable back to 3 nodes, and so we can calculate the distance. So basically 10 divided by 13, 13 nodes, 10 were the same. And we can test whether or not that difference is statistically unusual by doing a binomial test. And so here's lots of ways to do it, but in Excel or Google Sheets, you can do it by just simply equal binom.distribution 10. That's our in common 13 out of the possibilities. So this number is, you have to guess, well, what's the random chance of success? And so I just took 1 divided by 13 as an estimate. And 0 is just the type of probability we want to result in. And so it'll give us a p-value. And as you recall from your basic statistics, if a p-value is less than 5%, we'll tend to say that the gene tree is different from our consensus. Not a lot different. You know, remember, it was only 3 nodes, but there's some differences. We're not going to do anything. We can't fix it, right? But this is about doing basic science. So we're going to apply some analysis, and to be confident in our analysis, we need to know if our assumptions that are used, the models that we use, are based on assumptions that we have to make. And one of them here would be that this particular gene that I use is not very good at reconstructing the true phylogeny. So our assumptions are not holding there, and so we'd want to be a little careful in some of our conclusions that follow. All right. So what we're introducing is this model called a molecular clock. And so for a variety of reasons, we can expect that many proteins evolve over time, roughly proportional simply to the mutation rate, so that there will be a rough constancy of evolutionary rates. And if you make a plot of divergence times, so humans and chimpanzees last shared a common ancestor about 6 million years ago, for example. So that's the divergence time, 6 million years, versus the distance. So looking at your gene tree, what's the genetic distance between humans and chimpanzees? And if you collect that for a whole variety of pairwise differences, humans versus chimps, humans versus macaques, humans versus mites, et cetera, you can calculate this as a difference, and you'll find, where's my slide? Yeah, okay, that you'll find that over time, the more difference there is between 2 sequences, well, that correlates to those 2 species, diverge from each other a long time ago. Okay. So this slide's a little bit out of order. So the other thing I asked you to do was to calculate another test of the assumption, which is that rates of evolution throughout the tree are more or less equal. And so we can calculate the average rates of evolution, their lambda is the distance, times 2 times t, where t is the amount of time between them. Okay, so if you take 2 species and you compare them against an outgroup, which is shown in the Lab 8 worksheet, but it's not shown here, the expectation is that the distance between these 2 will be the same, indicating equal rates of evolution. Okay, so I have a worksheet for you to calculate that out. Okay, and it's an assumption. All right, so at any rate, so what we're going to end up with is that each one of you is doing a different gene product. And so you're going to be calculating your rate of evolution, your molecular clock, for your product, and then you're sharing your information with me. And in the end, I will provide you guys with a graph that looks like this. So all of your proteins will be plotted against time since divergence, and then the genetic distance, and you'll be able to take a look at your rate and see whether it's high or low. Okay, and so remember, high rate of evolution is consistent with the idea that chance mostly has been responsible for the evolutionary change in your protein. In other words, we would conclude that that's probably a non-essential gene product. And so unlikely that differences in that protein are important. Differences in that protein explain why one person has a disease and another person doesn't. On the other hand, slow rates of evolution suggest natural selection is purifying or preventing mutations from changing the protein sequence. And so we would think that this, in effect, is a marker of an essential gene. And changes to an essential gene, if they show up in our genome-wide association tests, then that is additional evidence to suggest that that correlation may, in fact, be causal. So low rates of evolution, essential genes, fast rates of evolution, non-essential genes associated with probably that an original correlation we identified, not an important one. Okay, so this is an example of some of the results that we've gotten in the past. Okay, so some of these are pretty wild, so these are pretty large, and so this is probably a reflection of the gene tree doesn't represent the consensus tree very well. Okay, but that happens. Not all genes are going to give us the same information. Okay, others are going to be showing us some pretty interesting things. And so this real big difference between humans and chimps, well, that suggests something special between us and them. Okay, so anyway, that's the idea about what this lab's about, the molecular clock. And so I'm going to go ahead and end this, even though there's more to do, but it's now coming up on 3 o'clock. Time for lab.